Rodrid. English is an infectious disease physician. He's the editor-in-chief of a journal named Journal of Infection. And I've been really impressed once I was with him on commenting what's new infectious disease by how broad his knowledge in the field was, which is something that is not very common. So he had a very broad spectrum vision of infectious disease, and this is why I want him to talk, but he preferred to talk about his experiment dogma, which is what he wants. But he will do that with this really broad vision that very few people get in infectious disease as well. So we're very happy to get him here.
Merci, Didier. Well, good afternoon, ladies and gentlemen. I'm very pleased to be invited to come here. Can you hear me at the back? Is that okay? And you don't mind if I speak in English. So here are your questions. This is the test. The questions are true or false. Up to 55% of English students carry Neisseria meningitidis in the nasopharynx. Most of those strains that are carried are not potentially virulent. Neisseria lactamica Carriage, that's a commensal, induces bactericidal cross-protective immunity against Neisseria meningitidis. Vaccination with the quadrivalent conjugate vaccine reduces serogroup B carriage, and inoculation of lactamica into the noses of volunteers, humans, modifies meningococcal carriage by gross alteration of the microbiome, true or false. So I hope that you can answer these questions at the end. So I'm going to talk to you about meningococcal disease because this now is relatively uncommon in Western Europe, with a disease rate of about 1 per 100,000 per annum. But when I was a junior doctor, this was a really big problem, because in the United Kingdom we had a lot of young people dying of meningococcal disease. We had an attack rate of about 3 per 100,000 and a mortality rate of about 20%. So it was not a good situation. And that's when I started to develop my interest in this disease. Now, some microbiology. When we survey this organism, we look for a number of biological phenotypes, notably the capsular polysaccharide serogroup. So, as you know, we describe Neisseria meningitidis as serogroup A, B, C, W135, X, and serogroup Y. And these are shown in these colors here. So blue is serogroup B, and yellow is serogroup C, and red is W135. But in the field of Neisseria biology, now when we want to describe the epidemiology, we are more interested in the sequence type, which is based upon the multi-locus PCR of 7 housekeeping genes. And in Europe, the most common sequence type is sequence type 41, followed by sequence type 11. Now, these sequence types tend to express either one or another serogroup. So, for example, in Europe, most sequence type 41 strains are expressing serogroup B, the alpha-228 polysialic acid capsule. But as you can see, different clones can express different polysaccharides. This is a panmitic organism, and it is quite promiscuous in uptake of genetic elements. So it can be transformed in nature to express one or other of these capsular polysaccharides. Now, it's quite interesting that actually, these sequence types are quite different in terms of the disease that they cause. For example, this one, sequence type 11, which in Europe mainly expresses serogroup C, has the highest death rate. Okay, so, and this is consistently seen. This was the sequence type that caused all the disease and death in England during the 1990s, and this was the reason why we introduced the serogroup C glycoconjugate vaccine. Also, this sequence type can express W135, and in Africa, about 6 or 7 years ago, there was an expansion of W135 expressing sequence type 11 organisms, causing a high death rate and bacteremia in South Africa and other parts of Africa. And it is this sequence type actually that is also associated with the Hajj in Saudi Arabia. Okay. Here's our view of the pathogenesis. This organism its natural habitat is the human nasopharynx. So we transmit the organism between each other by airborne droplets, and it inhabits the human nasopharynx. It does not, as far as we know, naturally occur in animals. We can induce colonization in primates, but it is very difficult to induce colonization of guinea pigs or rats. So there is not a very good animal model of Neisseria meningitidis carriage. So this is where this organism wants to be. So it's completely adapted for human colonization. This event, moving from the nasopharynx to the blood, is the pathognomonic step in meningococcal disease. And when it gets into the blood, it causes all of the features of the disease that we associate with Neisseria meningitidis. But this is a rare event. And we do not understand why this occurs. Because this organism is very sensitive to serum containing host defense molecules like complement and antibody. Okay, so what about some epidemiology? So if you take the general population, about 20% carry Neisseria meningitidis across the whole population. So that means that in this room, maybe 20 of us are now carrying Neisseria meningitidis. So look at your neighbour. They may be carrying the organism. But disease is, as I say, extremely rare. So although there is a big reservoir of carriage, disease only occurs in France. It is about 1 per 100,000 per annum. And mortality across Europe is always about 8% at the moment. Okay, during the 90s with ST11 it was much higher. So, I'm going to show you a lot of unpublished data. Alright, so I'm sorry about that. Most of the data I'm going to show you is very new. I'll show you some old stuff as well, but most of it's new. This is unpublished. So we did a big study in the UK involving 10 centres, and we looked at Carriage during 2010. And what we found was that our baseline Carriage among students was 33%. But this was very variable. In Bristol it was 20%, but in Nottingham it was 60%. And as the term progressed, as the semester progressed, the carriage rate rises. So when students get together, they socially interact, and their carriage rate rises. And when we did genotyping of these organisms, and this, as you know, there's a difference between serogrouping and genotyping for capsular operons, because an organism can have the capsular operon but may not express the polysaccharide. When we looked at the rate of capsulation, what we found was that less than half of the organisms were non-groupable. Okay, so not only could you not serogroup them, but they didn't actually have known capsular operons according to PCR. Whereas we also found that there was a lot of carriage of serogroup Y, and most carriage of capsular strains was serogroup B. Now, if we had done this 20 years ago, this would be serogroup C, but because we vaccinated, we got rid of serogroup C in our carriers in the UK. Now, when we look at just the serogroup B organisms, and we ask the question, are these disease-causing strains or not? The answer is yes. They are almost all of sequence types that have the potential to cause disease. So this means that the majority of the organisms that 20 of you are carrying, probably just over half of them will have disease-causing potential. They won't be benign strains entirely. Okay. Now, the reason why that is important is because of this slide. This shows the likelihood of causing disease if you are carrying organisms, Neisseria meningitidis in the nasopharynx. And this shows the data for the 1980s and for the 1990s, and it gives you these rates of disease given carriage according to year. And as you can see, during the 1980s, when we had a problem with sequence type 11 that was expressing serogroup C, even when we had very virulent organisms, the likelihood of disease given carriage was less than 1 in 1,000. Okay? So that's a surprise. Why is that a surprise? That's a surprise because this graph shows the rate of, when we measure antibody in the blood and we look for serum bactericidal antibody titers, we know that a titer of 4 or higher is protective against disease. And babies, infants, have very low titers, but the rates rise as we get older. And when we see teenagers, young adults at age 18, almost 50% of them have protective antibodies in the blood. So at least half of the teenagers have protective antibodies. So why is it then that these people are carrying very promiscuously strains of Neisseria meningitidis that predominantly have the potential to cause disease? Only half of them have protective antibodies, according to everyone's agreement about the surrogate markers, and yet the disease, given colonisation, is really rare. Okay, so that's a background for you, and I'm now going to talk to you about work that we did a long time ago, and which we still do in my group, where we examine this process of colonisation to try and understand exactly what it is. Now, this is a human nasal turbinate. So this has been removed from the nose of somebody with an obstruction of their nasopharynx, non-allergic nasal obstruction. And what we can do is create organ cultures derived from this tissue, and we can examine it in a petri dish, and we can infect the tissue directly, and we can perfuse the tissue with medium. And when we do this, we can examine the biology of this process of infection. And this is old research that we published a long time ago, and it generated some interesting information. So, for example, this is Neisseria meningitidis, so this is the surface of a cell, and the cell responds to the presence of the organism by producing these microvillus processes. So there's a directed response against the organism. And what happens then is that the organism is taken up into these parasitophorous vesicles. And as you can see, there is more of these extensions here. And it can live inside the cell at very high densities. And as you can see, the organism is dividing, it's very happy. Très content in the cell. So this is a non-professional phagocyte. This is very unusual behavior. When you look at other organisms that inhabit this tissue, none of them do this. So Haemophilus does not do it. Streptococcus pneumoniae does not do it. Just Neisseria meningitidis has this process of going inside the cell. Very interesting. And then if we look a little bit deeper, here is the surface. The organism comes through and between cells, and eventually, when you follow this process, this is the basement membrane collagen here. Here we can see that the organism has now come through to the basement membrane collagen. And it should see very soon an endothelial cell, and then it has then access to capillary blood. But this cell, which you can see here, this is a macrophage, and you can see that this cell is sending out these pseudopodia to phagocytose Neisseria meningitidis. It's also phagocytosing these apoptotic cells, but there is clearly a directed activity against Neisseria meningitidis. So, about 10 years ago, we worked out a system just to measure how well the organism survived in this tissue. And what we did was we used an automated, we call it a French press, okay? A press française. It's a machine that essentially we can take the tissue that I just showed you and we can turn it into liquid. And this just allows you to do steam age microbiology. So you can measure the organisms that you have infected your tissue with, and you can do quantitative counts between one treatment and another treatment. You can infect the tissue with Neisseria, you can knock out a gene, you can put in a gene, you can measure the effect of those changes. And this has been a rather useful way of measuring the success of these organisms in the tissue. But what we found was, and this was a piece of work that was published a long time ago, what we found was that the different donors, so each different donor handled the tissue very differently, handled the organism very differently to other donors. So I may take a turbinate from you or a tissue from you, and if I put Neisseria onto it, it's very different how this organism then responds to the presence of the epithelium. So we can see this is a log scale of the counts, the viral counts of Neisseria in the tissue, and this is at 4 hours and 18 hours. And what you can see is that there is a wide scatter of recovered viral organisms from the tissue. And this is certainly also true when we just look at the numbers of organisms inside the tissue. And this reminds me of this work. It's suggesting that people vary in the way that their tissue responds to Neisseria meningitidis. Jeffrey Rake worked in Boston, US, during the 1930s, and Jeffrey Rake ran a lab in Boston which just did meningococcal microbiology. And the reason for that was they had a massive problem with meningitis in Boston in the 1920s and 1930s. So they established big departments, and this was one. And what he discovered was this. All of these people were working intimately with Neisseria meningitidis, and they were all mouth-petting, pipette avec la bouche, because that's what they did then. And what they found was that there were some people who were never colonized by Neisseria, never colonized at all. No throat swab was negative, nasopharyngeal swab negative. And yet there were some who were chronic carriers. They carried it all the time. And there's no doubt that all of these people will have been immune. They will have had antibodies to Neisseria. So people are very different. And this story is a little bit similar to the story with Staph aureus. Okay, so it's an interesting observation. And I don't think that anybody has yet explained why this occurs. Why is it that some of us in this room will not carry Neisseria meningitidis? Okay, so using these techniques, we've learned a number of things. We've learned a number of things about the gene products of Neisseria that will enable the organism to penetrate and survive in the tissue. And here's a list of the gene. It's all a bit boring, the burn stamp collecting, but probably around about 10 or 15 gene products have a role in this. And we did much of this work in association with my friend Christoph Tang, and we used a technique called signature tagged mutagenesis, and we were able to screen libraries with this model and discover these gene products. So all of them have an explicable association. So the capsular operon greatly affects how well this organism does. Opacity proteins, these are capable of inducing the directed response. Pilin, obviously, because pilin is critical for adherence to the surface of cells. Lactoferrin binding protein allows the organism to scavenge iron. TON-B allows the organism to make bacteriocins. And lactate permease is a gene that allows the organism to capture 2-carbon substrates for its metabolism. And we also recently discovered that this organism can use nitric oxide to respire under microaerophilic conditions. So this is a very interesting fact because this tissue has a lot of nitric oxide. It makes most of the nitric oxide in your respiratory tract. And this is a bactericidal molecule. But this organism has a gene which allows it to use nitric oxide. Okay, and then there are some host molecules that are also important in this process. We know that the carcinoembryonic antigen receptors, these are critical for recognizing Neisseria, and so are the heparan sulfate proteoglycans series to which these organisms combine. And we recently showed that the tetraspanins are involved. Okay. So just to summarize, this organism can colonize the nasopharynx, it can come through the tissue, but this cell is the macrophage, and this is trying to kill it. And I told you that even though 20% of us are colonized with potentially virulent organisms, only 1 per 100,000 of us get disease. Despite the fact that less than 50% of the people in this room have protective antibodies in their blood. So what is stopping this? Well, the answer is probably the innate immunity of the host. It's obviously critical. And so we used human monocyte-derived macrophage, these cells, to answer many of the questions that were raised by this. And we find that there are a number of factors that are very important. The first is that clearly the organism's expression of capsule is critical. And the more capsule it's expressing, the better it is at surviving macrophages. And we know that although the organisms in the nasopharynx predominantly are down-regulating polysaccharide production, the ones in the blood are up-regulating it. And so it's very clear that this product is critical for surviving macrophage assault. And there are a number of other factors that the organism have that allow it to resist macrophage killing, including IgA protease, lipopolysaccharide, and 4B, another product. Okay. So, I'm now going to talk about another model that we created to try and study colonization, because we were very interested in the fact that there is a relationship between Neisseria lactamica and disease caused by Neisseria meningitidis. So in this study, which was done in a little village in England called Séminaire that had a terrible, terrible disease rate in the 1980s, they did a colonisation, a carriage survey, and on the y-axis is shown percent carriage of the population that they surveyed. On the x-axis, the x-axis is showing the age groups. And what you can see is that carriage in Neisseria meningitidis rises during childhood, reaches a peak in the teenagers, and then drops off. These people, the 15 to 20-year-olds, they are where all of the carriage is. All right? So you can see that you're all mostly in this age range here. So you're also a bit of a reservoir of this organism. But lactamica, which is a closely related commensal, and it's really important because it has a 70% genetic homology with Neisseria meningitidis, and many of the genes are practically identical that it has. But it does not express a capsule. This organism is highly prevalent in infants, especially nursing infants. That's why it's called lactamica. And it drops off in childhood. So that we see very little lactamica carriage in adults. And then there seems to be an inverse relationship. Now, this was also replicated in the United States, where they showed, again, the same pattern of lactamica carriage, and these are babies, and as age increases, lactamica carriage drops off, and meningococcal colonization takes up. And this association between lactamica carriage has also been shown for disease. The Scandinavians have examined disease epidemiology in Iceland and Denmark and shown that where you have a lot of lactamica carriage, you have less meningococcal disease. So, we did an experiment. We wanted to know why this was happening. Because one thing could be that these people are making antibodies. Maybe they are making antibodies. Maybe if you are carrying Neisseria lactamica, you're making an antibody which cross-protects you against Neisseria meningitidis. So we wanted to directly answer this question because it's important. And the reason it's important is because we would then understand more about this biology, and perhaps we might be able to replicate this inhibitory effect of lactamica on meningitidis. So we wanted to create a vaccine based on Neisseria lactamica. So this is Carriage Evans, and this is a willing volunteer, and she is inoculating the volunteer with Neisseria lactamica, and this is a defined strain which has been sequenced and about which we know a lot. And we asked the question, well, how long does carriage of this organism last? So when we put it into volunteers, how long does the organism stay there? And what we found was that this shows the percentage of volunteers who are carriers is that we managed to achieve a colonisation rate of 60%. So 6 out of 10 of the people that we inoculated could carry lactamica. And the number of organisms we put in was 10,000, 1 times 10 to the 4. And this carriage persisted for 12 months. And I should say that I was the first person to be inoculated with this organism, because I was the chief investigator and I wanted to make sure it was safe. So we then asked the question, does that induce an antibody response? So when we inoculate Neisseria lactamica, does the host make an immune response? And the answer is yes. So shown here is the salivary IgA concentrations measured against the standard. And what we see is in red that when you are colonized by Neisseria lactamica, when we induce colonization, there is a rise in salivary IgA, which is significantly different to controls. Interestingly, there's a drop in those people who are inoculated but are not colonized. And when we look in the serum, we see the same thing. This is the serum IgG done by ELISA against the precise strain that we have inoculated. And again, the people who were colonized are in red, and there is a dramatic increase from 2 weeks of serum IgG against this organism, which again is flat in the comparators. So, they are making an immune response. Actually, this was one of the first direct pieces of evidence that carriage of a commensal will generate an immune response, which is quite important. But then we ask the question, well, does carriage of lactamica induce a cross-protective immunity against Neisseria meningitidis? And the answer was no. So this data in black is the people who were colonized, and in white are the controls, and in hash are the people who were not colonized. And this is the percentage of people with serum bactericidal antibodies against strains of Neisseria meningitidis, serogroup B. So these are different strains of serogroup B Neisseria meningitidis. And what you can see is there's a little bit more amongst the people who were colonized, a little bit more serum bactericidal antibodies were made. But this rate is pathetic. It's really low. There's a bit, when we look at opsonophagocytic antibody, done using standard flow cytometric assays, then we do see a little bit of an impressive cross-protection. But this opsonophagocytic antibody is of questionable importance. So, the answer is, really, people do not make an impressive immune response to lactamica, which protects them against Neisseria meningitidis. So why is it then that people who are carrying Neisseria lactamica are protected against Neisseria meningitidis? If it's not immunity, what is it? Well, we did this experiment, which was to inoculate again a huge number of students and young people with Neisseria lactamica. And this time, we measured the carriage of Neisseria meningitidis over the course of 1 year. So we did serial throat swabs on all these volunteers, and we had test and control, and we followed their carriage. Now, at baseline, this shows the rate of Neisseria meningitidis carriage. Now, this time, the carriage rate was only 23%. And this is because we chose only non-smokers. And there was no difference between the people that we challenged and the people who were controls. And then we measured Neisseria lactamica colonization. So we inoculated the organism, and we found this time that we could induce carriage of Neisseria lactamica at a rate of 34%. when we divided into those who were not colonised by lactamica and those who were colonised, we just checked whether or not we managed to put the same amount of lactamica into the two sets of volunteers. And what we found reassuringly was that there was a very tight comparison between the inoculation of people who became colonised and people who did not become colonised. So it was not an experimental artefact. And this graph shows lactamica carriage in the volunteers over the course of the experiment. And here is showing the rate of lactamica carriage. So at baseline, lactamica carriage was only 0%, and then we inoculated them, and then in the challenge group we got a rate of 34% by 2 weeks. And this remained stable over 6 months. We then re-challenged all volunteers at 6 months, and we got these rates. All right, so again, we managed to re-inoculate at the same rate. So, this graph shows Neisseria meningitidis carriage. So, this time, again, the challenge group is shown in red, and the control group is shown in blue. Here is shown the rate of meningococcal carriage over the course of the experiment, and what you can see is that there is more meningococcal carriage in the control group compared to the group that we inoculated. And this is highly significant. But when we look closer at the data, what we see is this. This time, in hashed bars are shown the people whom we challenged, and then we detected Neisseria lactamica. And in the full green bar is showing people whom we challenged, but we did not find Neisseria lactamica. So in other words, the hash bar are the people where there was induction of colonisation of lactamica. And now you can see there is a dramatic effect of Neisseria meningitidis on the carriage. There is a dramatic effect of Neisseria lactamica on the carriage of Neisseria meningitidis. And this persists for 6 months. Okay? It persists for 6 months. Okay, so this graph just shows this graphically for you. So this shows the control group, and this shows the challenge group, and in green, these are individuals. Across here there are 300 individuals, all right? And this shows the throat swab results at 0, 2, 4, 8, 16, 26, and 28 weeks. And green is lactamica is present, and blue is no Neisseria present. And you can see that in the challenge group, there's a lot of green here because we've put lactamica in, and it stays there for up to 6 months. And then at 6 months, we then inoculate again, and you can see that there's more green. In the control group, there's very little blue because we have not inoculated lactamica, and then we inoculate lactamica at 6 months, and there it is. Now, when we superimpose on this meningococcal carriage, now shown in red, what we see is less meningococcal carriage in the people we inoculated with Neisseria lactamica. And this shows you the pattern that we see. As you can see, over the course of 6 months, students have more meningococcal carriage, whereas in this group, this seems to have been interfered with. Okay, so this could either be because we are displacing the organism, or it could be because we are preventing acquisition. So this slide shows the displacement of Neisseria meningitidis. And what we did was we took people who were carriers of Neisseria meningitidis at the start of the experiment, and this shows the meningococcal carriage rate, and this is weeks after inoculation. And what you can see, in blue is shown the control group, and in red is the challenge group. And you can see that there is, although there is a trend, there's not a significant difference here. But then, when we just look at the people who are lactamica carriers, what we find is that there is a significant displacement of meningococcal carriage by lactamica. In other words, there's competition. What about acquisition? So this time we're looking at people who were not carriers of Neisseria meningitidis at the start of the experiment, And we're looking now at acquisition of Neisseria meningitidis. So again, the meningococcal carriage rate is shown here, and time is here. And there is a significant change across the whole groups across the 6 months. But this shows, again, in the dotted line, the people who were carrying Neisseria lactamica for almost 2 months had no acquisition of Neisseria meningitidis. So these are university students. So the presence of lactamica is stopping them from carrying Neisseria meningitidis. Whereas in the control group, they're all kissing, they're all interacting in bars, etc., and they are carrying lots of Neisseria meningitidis. Look what happens when we give lactamica. Nothing. They don't carry it. So that's an interesting observation. So it raises the following possibility. This is our population, and here is the colonisation with Neisseria meningitidis. And these are the hypervirulent strains that I've told you about that we don't want people to carry. Maybe we can remove hypervirulent carriage one day by changing the biology of the nasopharynx so that people tend to carry benign Neisseria meningitidis. Okay, so the nasopharynx is a community. Bacteriologically, it's a community of thousands of bacteria. And the microbiome of the nasopharynx is not very well defined. But we need to know whether or not this lactamica interaction with meningococci was specific, or whether it was just a gross alteration of the microbiome. Does it just completely change the microbiome? So we took the throat swabs, we put them into PBS, and then at the Sanger Institute in Cambridge, they did 16S ribosome sequencing, looking for the microbiome. And I'm just going to show a few examples of the results of this. I mean, there are many, many different species. Here is shown the different genus that we managed to acquire during these experiments. So I'm going to show you data for 3 individual donors. The bottom line is that we did not see a consistent pattern. Everybody has their own microbiome. All right? I think we know that. But it's also true. We know it for the gut, but we now know it for the upper respiratory tract. And when we inoculated Neisseria lactamica here, and this has shown the change in the microbiome over a few days, and what you can see is that there's a fairly consistent pattern. This just shows the percentages of each of these genus in this individual. And here's the donor. This time, in red actually is Neisseria meningitidis, and lactamica is in dark purple, but you can't see it. We've only put in 10 to the 4 Neisseria lactamica. You know, we recover 10 to the 3 from nasal washings, tiny numbers compared to the rest of the organisms present. And as you can see in this donor, we had Neisseria meningitidis, and then by 2 weeks it's gone to nothing. But, you know, there's not really much of an alteration otherwise in the rest of the microbiome. In contrast, here's a completely different donor where the opposite occurred. We've given lactamica. Lactamica has not grown, but they've become colonized by Neisseria meningitidis, and it wasn't present at the beginning of the experiment. So, and as you can see, across 4 weeks, not much change at all in the microbiome. No dramatic effect. So, we've inoculated Neisseria lactamica. We have deleted in some people Neisseria meningitidis, okay? But despite this very dramatic effect, there's no change in the overall microbiome. So it's not being done by modifying greatly the relative proportions of other genus in the nasopharynx. Okay, so I've got 5 minutes, I think. So in 5 minutes, 10 minutes maybe, I'll just talk about another very, very recent experiment that we did that will be published later this year. We're still on the same theme. We're on the same theme of carriage of Neisseria meningitidis. Now, as a background, in the UK, when we gave the Men-C vaccine, we started in 1999, and we did it because in 1995, we had a huge number of cases of Neisseria meningitidis serogroup C disease, and it was rising through the 90s. And I was working as a junior doctor at this time, and it was terrible. Every week you saw in my hospital 3 or 4 bad cases in the UK. And it was an expansion, as I told you, of sequence type 11. And then this is what happened when we started to give, in 1999, the serogroup C vaccine. And there was a dramatic effect on the number of cases. So now we never see cases. They're only imported cases. Okay? So there are only people who've grown up abroad and come to live in the UK. And Martin Maiden in Oxford, after this was done, he measured amongst 45,000 students in the UK. He did throat swabs, and he measured the carriage of Neisseria meningitidis. And what happened was this. When he looked at whether they were making serogroup B, C, W135, etc., there was a significant fall in the carriage of serogroup C. And that's important. And it's important because of this. So what he saw was a reduction in serogroup C carriage within 1 year. Now, these students had not been vaccinated. Okay? Some of them had, but not all of them. And indeed, when we look at disease rates, what we saw was a drop in disease, not just in the children, which is shown here in blue, but even in the older people, we saw a drop in disease by serogroup C. These people, in red, did not get the vaccine. Why has this happened? Well, obviously it's happened because the vaccine has interfered with the carriage of Neisseria meningitidis, and that's how it's produced this herd effect. So it's reduced the burden of carriage of Neisseria in the population and reduced the effect. So we did this study where we did a phase 3 multi-center randomized controlled trial, and this time we had a new set of vaccines. So recently you'll be aware that most of the disease, certainly in France, is caused by serogroup B. There is no vaccine for serogroup B yet. Okay? There's one licensed by the EMA. It's not been deployed widely yet. And there's also a new quadrivalent vaccine, conjugate vaccine, meningitidis, but there's another non-conjugate quadrivalent vaccine. And this is expressing ACWY. So what we did was we asked the question, well, if we vaccinate students and follow them for 1 year, do we see a change in carriage? And if so, how much by? How much is that? Because if we know exactly how much an effect it has, we can learn a lot about how to model the effectiveness of vaccines. And we were expecting this vaccine, Menveo, we were expecting this to have the same effect that I described that Martin Maiden showed for the Men-C. We expected it to change serogroup Y, W, and A carriage. But there's a new vaccine called Neisseria, and this is a protein-based vaccine that has been designed to be active against serogroup B. And that's because it contains these 3 recombinant proteins plus the outer membrane vesicles of H4476 strain, from which has been removed 4A, because this is an immunodominant epitope and you don't want it. And this vaccine was invented by Rino Rapulli in Siena. And is marketed by Novartis and is designed as a serogroup B vaccine. So what we did was, we took 1,000 people and gave them this new vaccine, a protein vaccine. We took 1,000 people and gave them a quadrivalent conjugate vaccine, and we took 1,000 controls. These people received 2 doses of Bexsero, and we gave the controls this vaccine, which is the Japanese B encephalitis vaccine, which should have no effect on carriage. And then the third group we gave Menvio. This is a single-dose vaccine, so they got placebo for the next dose at 1 month after the first dose. And what we wanted to learn was whether or not there was a change in the swab, the culture of the swab. Did we find more or less carriage in Neisseria over the course following the vaccination? So this is unpublished data. The primary analysis was 1 month after the dose, finishing the course, what was the carriage of Neisseria meningitidis, and the secondary analysis was the cumulative carriage in the months following this. For the protein vaccine, again, the primary analysis was 1 month after finishing the course, and the secondary analysis was the cumulative carriage in the periods after vaccination. And this data just shows the baseline meningococcal carriage. And so this basically shows the carriage rate before the student got the vaccine. And as you can see, it was a dynamic process because we were enrolling 3,000 people. So we enrolled them over these weeks of the year. So we tried to do the experiment where we just did it in 1 week. But unfortunately, that's impossible. It's not impossible, but it's difficult. But this just shows that over the course of the term, there was a rise in baseline carriage. And overall, we had 33% average carriage. And 9% were carrying Neisseria meningitidis serogroup B. So this shows the primary analysis. And essentially, there was no significant effect of either vaccine 1 month after the last dose. Okay? And then, so now I'm just going to go straight to the secondary analysis, because this is where the significant data is. This just shows, the quadrivalent conjugate vaccine, this shows the carriage of ACWI serogroups, all right? And what we now find, there is a significant fall in carriage of those serogroups when we give the vaccine. And especially true when we just look at serogroup Y. There was no effect on carriage of serogroup B because, of course, that polysaccharide is not actually in the quadrivalent conjugate vaccine. So, this data is showing us that the quadrivalent conjugate vaccine has a significant effect of carriage on ACWY over the course of 6 months. So, this would have a dramatic effect on the carriage of these disease-associated serogroups. So, in France, if you had an expansion of highly virulent serogroup W, you could use this vaccine by targeted deployment to remove disease very quickly from your population. What about serogroup B vaccine? Well, what we found here was there was an effect. It was a smaller effect, but it was still significant. And certainly across all the parasitic groups, there was overall a fall in carriage. This is with a protein-based vaccine, so this is quite a dramatic observation. And when we looked at any Neisseria meningitidis, there was also a fall, but this vaccine was designed to protect against serogroup B, and here we saw no significant effect. Okay? So there was no significant effect on the carriage of serogroup B. Mind you, please note that the baseline carriage rate was only 9%. So what this means is that this protein-based vaccine can cause a reduction in Neisseria meningitidis carriage. But please note that we do not know yet whether that reduction would have a beneficial or a harmful effect. Because these strains that it's reducing, they could be protecting the population, for all we know. Okay, I'll stop there. So, I would just like to thank my contributors. There are many and varied. Here are the folks that helped with the vaccine study that I just showed you. But I particularly want to acknowledge these individuals who helped with the lactamica challenge, because if you've ever done an experiment where you've taken humans and you've infected them deliberately, I can tell you that is a difficult thing to do, and it's very, you lose coronary arteries. But here is the woman who ran that study for me, Alice Deasy, and she will be the first author on the paper when it comes out later this year. So thank you very much.
Again, question, do you test if Neisseria lactamica got a specific bacteriocin to Neisseria meningitidis?
Yeah. So the question was, does lactamica have bacteriocins? We've done a very crude experiment and we cannot see that. But of course, we've only looked at a couple of strains of Neisseria meningitidis, and the bacteriocins can apparently be quite specific for the targets that they will affect. So we have not done a satisfactory screen. We do have all these strains, so we are in a position to address that question. What we do know is this. We took the baseline strains of Neisseria meningitidis, and we were able to differentiate the ones that were displaced and the ones that were not displaced. And when we just did simple broth culture experiments, what we found was that the growth curve was very different between those two groups. So the ones that were displaced actually had 1 log differences in growth, over 18 hours. So there is a difference, apparently, metabolically, between the meningitidis that seem to be vulnerable to lactamica and the ones that do not seem to be. Now, that just tells us that they may be vulnerable to competition. Whether that competition is bacteriocins, or whether it is nutrient competition, or whether it is a phage, I don't know. We've looked for phages, we can't find phages. But again, our techniques are primitive.
Yeah, it's interesting because we're quite close to this on trying to understand how lactobacillus is changing the microbiota of the gut.
Of the gut, yeah.
And it's been quite tricky to find. And we was good idea. Maybe you could meet him at the moment. After we've used a new test to make a kind of antibiotic susceptibility testing by using the strains that we want to test against the other in agar. So it's going to be a little tricky, but some of the tests that have been done several years ago are close to this. And obviously there is. It's a good way to screen specific bacteriocins, so there's very specific species, which is maybe one of the best ways that bugs are controlling themselves.
Yeah. I think the technology that we're using is probably quite primitive, so I think it would be good if I can find out what technologies you use here, because I think I might need to find out more about this. We have looked. We can't find any so far. Just simple lawn techniques.
And the other thing, I'm ignorant of what is the capsular antigen. I understand very well that you get a clone that could have a capsule of B or C capsule. What is the genetic support of the capsule? Is that transponding or what is it?
Okay, so it does appear that quite large segments of the genes are deleted and then replaced by something else. I'm afraid I don't understand the mechanism in which that occurs, but I know that Richard Moxon's group have gone some way to try to define how that happens, but an entire gene has been moved in, and the entire gene has been moved out. Now, presumably the organism decides to delete the old gene in favour of the new one. But the technology, I don't understand the mechanism of that. So
Does the same of salmonella, for example?
I don't know.
Questions?
No, almost time.